Hello, everyone. Uh, Thank you for coming in this afternoon. And I know you just had lunch, so I appreciate your patience. So this afternoon, we're going to get started with the Neuromorphic Engineering for Clinical Care Minisymposium, as organized by myself, as well as Grace Huang and Joe Monaco from the NIH Brain Initiative. Grace and Joe wish that they could be here, but due to the whole government shutdown stuff, it was a little difficult for them to make it here. So I'm running the show in their stead. So we're all in this together. All right. So thank you. So I guess in case you're wondering why the NIH is interested in neuromorphic computing and why they're kind of setting up this session here at this conference. So we think that right now that this is kind of a convergence moment for neuromorphic computing. And why now? So in particular, there's a data revolution, right? So the Brain Initiative, a big funding initiative through the NIH, NSF, and various other US agencies, has created unprecedented neural data sets, such as complete connectomes, which you'll learn a little bit more about how those are being mapped to neuromorphic hardware very, very soon. Various cell atlases and connectivity maps. Right. Additionally, at the same time, AI is starting to hit the wall of cost and energy for training. Right? So training these large-scale, large language models is estimated to cost about 100 million and 100 gigawatt hours, enough to power a small city for a year. Right. So very unsustainable. But at the same time, neuromorphic chips are starting to demonstrate that brain-like efficiency can be achievable at brain scales of various different types. So yeah. And so we think that this is a perfect storm for synergy. Right? So this convergence is creating a unique window for transformative progress. So the talks that are going to be given over the next 90 minutes or so are going to be showcasing different ways that neuromorphic engineering can further neuroscience, as well as neural engineering and different aspects of clinical care. So with that, I'm going to get off the stage and we're going to welcome up our first speaker.
All right, well, thank you, Will. So I'm Brad Aimone. There you go. I'm a researcher at Sandia National Laboratories, and I'm kind of excited to be part of this Brain Initiative Minisymposium here at this conference. What I normally talk about is the application of neuromorphic computing to scientific computing and sort of HPC and those sort of domains. But what's coming more of a reality, and we'll kind of set the table for this, is that the Brain Initiative over the last, decade has been collecting neural data at scales that is unprecedented. And we believe that neuromorphic computing may be able to kind of recapture some of that initial promise to help deliver deeper neurological insights about how the brain functions. And most importantly, particularly for this community, the ability to actually start treating things. So I want to motivate this a little bit, kind of going back to basics of biology, right? We know that from the central dogma of molecular biology, there's this idea that you have a genome, and that gives rise to the so-called transcriptome, which mRNAs are created from the DNA. And that gives rise to the proteome, which is the proteins that are translated from that mRNA. And this model has been around for 75 or so years, going back to early studies of DNA. And this underlies almost a huge amount of our ability to clinically address major diseases, things from cancer to heart disease to infections and everything. We don't have this for neuroscience, right? Our treatments, insofar as we have them, tend to be somewhat ad hoc from a theoretical point of view. And that's not good. We want to get past that. So how do we, the brain does computation for our bodies. So how do we treat the disease computation? So I just want to, kind of push on this a little bit, right? 5 years ago when COVID came out, it was kind of like, oh no, what do we do? Well, molecular biologists, because they're awesome, were able to sequence COVID and target particular proteins on it and create vaccines in 6 months. So basically, having this sort of strong theoretical framework and the ability to technologies to actually go in and look at the genome and push forward allowed a revolution that helped bring the world out of crisis. And we would love to have this for neuroscience, right? And I think this is a goal that's not near term, but it's something that's within reach. So just want to pay attention to the very bottom statement here, right? And I think this is the key thing that we miss in neuroscience. The transcriptome is constrained by the genome. The mRNAs that our cells produce have to be encoded by the DNA in those cells. And it kind of goes without saying. And the proteins that are produced have to come from the mRNAs that are produced and back to the genome. So what is the equivalent of this for neuroscience? And Will mentioned the connectome is something that has kind of come along online at scale in the last few years. The FlyWire connectome, which you see on the bottom left here, is the fruit fly brain. It's a few 100,000 neurons, many orders of magnitude smaller than the connectome of a human brain. But it's worth pointing out that when the first genomes were made of systems in the 90s, within 10 years, we had a human genome. And that first genome cost billions and billions of dollars to produce. And now you can sequence someone's genome for a few $1,000s, right? Now, connectomes are different, but nonetheless, it's reasonable that technology, when it identifies a target, will scale to address it. So how do we get this idea that we can constrain activity and computing from the connectome? So I'm going to throw out a hypothesis here. It's mildly provocative, depending on the audience, that neuromorphic computing can help facilitate this. In particular, the ability to simulate the brain and understand what the brain is doing, as well as the ability to identify the sorts of computations the brain can do. So Will showed this slide earlier. We now have neuromorphic hardware, the digital kind of standard fabricated type, that's getting up to billions of neurons. Analog hardware, which you kind of see on the bottom right, is scaling as well. It's just a little further behind, but it's getting there. And what's important to note is that this hardware is reaching scales comparable to that of biological systems that are important from an experimental point of view. So right now, if I were to simulate the human brain on a supercomputer, I would need an exascale machine to simulate the 100 billion neurons and the 10 to the 15th synapses. But it's not unreasonable to think I could build a neuromorphic supercomputer that could do this far more efficiently. And I'll show this in a few minutes. So we wanted to do a proof of concept of this. Sort of the first thing I'll show is that we were able to take this FlyWire connectome. So part of the series of papers, they were Brain Initiative led. We weren't involved. We're not fruit fly people. But they had a series of papers describing the connectome. And one of them was a very nice computational model of the full circuit. And so we took this paper, you can see it on the bottom left there, and we took the model, this kind of a standard leaky integrated fire model, if you're familiar with the term, and we deployed it on our Loihi 2 test beds, the neuromorphic chips. Each chip has about 1 million neurons on it. And so we, these little boards, we had them out in the front room there the last couple of days. So what we can see is that these chips are able to simulate the hardware, and this is what you'd expect. That's what they're built to do, but no one has ever actually had a real brain put on a chip before. And it turns out that this works. This paper is, we're preparing it for submission right now. It's on archive if you want to look at it. And what's important is that the simulation is replicating the activity. There's stochasticity in it, so the exact spike times aren't exactly the same, but the neurons are doing what they do in a normal simulation. So the neuromorphic hardware can replicate, from a simulation point of view, a high quality version of the models that we do on CPUs and GPUs. So the neuromorphic hardware is there. And more importantly, it's doing it orders of magnitude faster. And this isn't that big a deal for 100,000 neurons. I can still do that simulation on a laptop. But if I want to do a human connectome or a mouse connectome, the Allen Institute has a visual cortex from a mouse. We're getting to scales that, if I'm going to do sensitivity analysis, parameter sweeps, and so forth, I'm just not going to be able to do without a supercomputer. But if I have some neuromorphic hardware, it becomes a viable strategy. So this says that neuromorphic hardware can help take this connectome idea, which is kind of a static view of the anatomy, and turn it into something dynamical that we can then compare and integrate with the experimental results coming out of the neuroscience field. So then what do you do with this? And so I'm going to segue a little bit to a non-neuromorphic per se story that's motivated by the ability to look at the scale, where we want to start asking, how do we get past this big data challenge, right? And maybe tie things towards computing. Because our ultimate goal is computation. The brain is not just active for the sake of activity, but rather is trying to solve problems. So we can do these big simulations either in CPUs, GPUs, or neuromorphic. But what we'd really like to do, and there's a poster on this by my postdoc, Brett Thielman, at SFN next week, if anyone's around still on Wednesday. We would like to be able to get away from this idea of looking at the activity of neurons as like a static snapshot. These neurons are active at this time. These neurons are active at this time, but rather have this like dynamical view. Now, this crowd's probably going to appreciate dynamical views of information processing. That's not a common thing in neuroscience. There's a view, kind of a growing view of it, that we need to look at manifolds and other things. But for the most part, we're limiting this idea of here's a snapshot of firing rates. But in fact, it would be nice to say that there are trajectories of neurons that are kind of working on top of each other. And so we have this activity, this process, this paper's on arXiv that's called NATS. And the idea is that we can actually graphically decompose this sort of what we call a raster plot, all the activity of neurons, and we can constrain it with the genome, right? And so using this sort of formula you see on the bottom right, is basically a causal analysis. But instead of looking at like Granger causality and what neurons correlate with others, we're actually asking which neurons correlate with each other, constrained by that connectome. Because we were handed this connectome now. Now we have the ability to look at what the circuit diagram is. And that helps shape what the computation is. So what's nice about the NAT analysis is that we can see that you see the same patterns occurring over time of the same input. So this might be an idea. We're confident that this is actually something that could be a computational basis, in a sense, kind of kernels or motifs of dynamics that one can build algorithms out of. And this is something, if you kind of think of a sensory motor sort of transition where you had these temporal blurring of things, that we actually think that we can start building algorithms up from. So this is a bottom-up approach of how do you extract algorithms out of a connectome. But that's still not the answer. That's not telling us what the computations the brain is doing. And this, of course, is the, say, trillion-dollar question nowadays with AI evaluations, right? What is the brain actually doing and how is it doing it? So I don't have the answer, of course, but what I can say is that neuromorphic hardware might give us a different perspective on this than, say, what AI is doing later. So part of the challenge with AI algorithms is something called the universal function approximation. You take any neural network, it can do any function. So the fact that a neural network can do something doesn't clue us into what the brain might be doing. But one thing that we've done from a scientific computing point of view, which kind of motivates a lot of our work at Sandia, is we can ask, here's some physics problems, and can we map physics problems onto neuromorphic hardware? And I won't, we only have a minute left or so I won't go into great detail on this, but basically, if you take the sort of weak form of a partial differential equation that you kind of see on the top of this, where you describe your partial differential equation as a sparse linear system, something that's very important for things like finite element simulations and kind of understanding the dynamics of things like climate models and so forth. We can actually map that directly into a neuromorphic circuit. And this is a circuit that's based on the motor cortex, actually going back to a paper from Sophie Deneve about 15 years ago. This paper just came out today. It's in Nature Machine Intelligence, so I encourage people to look at it. Well-timed. But a neuromorphic circuit that is actually shaped directly by this sparse linear system. It's not learned. We just map the physics equations into the neuromorphic hardware. And it exhibits dynamics that you might look at and say, oh, that looks kind of like what neurons should look like if we're recording from things in vivo. And if you look at the sort of raster plots in the model, it looks exactly like the sort of things you might see in a motor cortex. And as you can see from the right, the model is actually fitting the physics data. The neural solution is fitting the physics data perfectly. Very high accuracy. And this is just some kind of information showing what it looks like compared to conventional solvers. For all intents and purposes, it's doing the same thing. We're doing straight numerical computing at HPC scales with HPC quality scaling at low power for neuromorphic. And this is a brain-inspired algorithm doing this. And it's something that the brain itself could very well be doing. And I would argue, and this is something that is not the normal way of thinking of neuroscience, is our brains have to solve deep, complicated physics problems just like our scientific computing codes do. If I'm going to catch a baseball or swing a tennis racket to return a serve, I have to solve these hard problems. I may not consciously be solving a sparse linear system, but very well, my neurons may be doing that at the circuit level. And this is something that we believe is actually a clue towards what the brain actually may be able to do. So with that, I just want to kind of go back to this sort of motivation where, if we can actually start using the connectome and use neuromorphic hardware to help push these constraints forward from anatomy into activity into computation, maybe it'll give us a foothold going forward to this more deeper impression about what the brain may be doing, and that may help us have a more strategic way of going about treating neurological disorders. So with that, I'd like to thank you very much. I appreciate your attention, and thank you to everybody who has disappeared from the slide. All right.
Hello. All right, so let's all thank Dr. Aimone one more time. So in order to keep this session moving along, we're gonna move on to the next speaker. But if you want to ask Dr. Aimone some questions at the end, I'm sure he's happy to take any questions that you may have. So our next speaker is Jason Ashragian from UC Santa Cruz. He is most famous probably for his Python spiking library SNN Torch, which he may pitch in his presentation. He may not. We'll see. And anyway, take it away, Jason.
Thanks, Will. G'day, everyone. I'm very relieved to not have to talk about neuromorphic language models in the next 12 minutes. But let's take a step back to when I was hired by UC Santa Cruz's faculty. Right? So this is a beautiful picture of the scenery of Santa Cruz County, about a couple of minutes away from my office. Now, when I moved from Australia to the States, I wasn't able to transfer my Australian driver's license, and it took me a couple of years to get to the DMV. And so I'd catch the bus and I'd see the scenery and I'd be like, God damn, this is incredible. That's day one. Day two, still incredible. Day 512, Jesus Christ, I got to grade all of these undergraduate exams for my analog electronics class. And the magic kind of disappeared a little bit, right? That's because my brain knew what to expect. So there's some sort of analog here to predictive coding, just in your day-to-day life. And so having this ability to suppress sensory information, I mean, on the one hand, it helps. On the other hand, it leads to anxiety of having to grade. But we're trying to use these ideas in order to, improve clinical care to some extent. So let's take a look at how this might be applied. I'll paint a picture for you. We're trying to develop new learning rules that take inspiration from these ideas. And we recently published one of them called Future Guided Learning. Applied it to a variety of different scenarios, and I think the seizure forecasting detection problem is an interesting one. So you have your patient, you're reading several channels, it's 24, 25 EEG channels, and you've got one model, a detection model that's trying to say, hey, is there a seizure happening right now? On the other hand, you have a forecasting model that's trying to say, is there a seizure going to happen in 30 minutes time, or whatever your time horizon is? Now, on the forecasting side, it's effectively going to enable preventative measures, but it's difficult, right? Telling the future is, of course, going to be more difficult than telling the present. But as far as detection goes, that's more or less as good as your model is going to get. So the idea here is kind of simple, right? If your ground truth isn't going to be as good as it gets, if your detection sets that sort of upper bound on how well your forecasting model can perform, then let's use the detection model to train the forecasting model. Let's project information from the future into the past in order to enhance your model. So let's kind of take a look at how this works. And I'll get you to click play on this animation. So you're moving forward in time. At some point, your detection model is going to ring an alarm bell. It's going to say, bam, there's a seizure. But the problem with this picture is that the forecasting model should have lit up 30 minutes ago, right? And so when there's a mismatch, you use your detection model to effectively update your forecasting model on the fly. And so that starts to look a little bit like a continual learning problem. And it turns out that there's actually a lot of benefits that we didn't really expect as a result of all of this, right? So there's no need for manual labels other than your initial trained detection model, right? is your detection model is effectively labeling everything for you with a forecasting model. As a result of that, you can effectively kind of sidestep the neurologist who's going to be very expensive. You can get the detection model to handle all of that for you. You can get a patient-specific model that, I guess, adapts to their neural signature, to their particular brand of epilepsy, if that's what you need. And as a result of all of that, you end up with a higher area under your curve, your receiver operated, your OR crop. Cool. Very messy graphs. We've got three baselines here. So along the x-axis, you've got a few different patients. On the y-axis, how good is the area under your receiver operating characteristic curve? That's kind of messy. Let's just average all of that out. There we go. Average all of that out, and in general, you're always going to get a performance boost across what was previously the state of the art insofar as seizure forecasting was concerned. And so I think that's kind of nice. The main takeaway here is that you really can minimize surprise by learning from the future. Now, in the neuromorphic world, a lot of us are very concerned about energy costs. And I guess that's why we're looking so deeply into, actually, I think the majority of my lab is looking at large-scale problems, much like Brad's. And so, yeah, naturally, we fall into the trap of working on language models. But in this case, how is it to train, right? Well, sure, you're only learning at mismatch. Whenever your detection and forecasting models diverge, then that's when you update. Otherwise, you're good. That could be a win. But on the other hand, if you're really trying to deploy this on some sort of edge device that is not going to be powered, but very power hungry, then you're trying to run multiple models at once. Bit of a pain, right? So we saw this coming. I guess over the past 4 or 5 years during the COVID era, I taught myself Python by putting together SNN torch. So I'll get you to click play on this cute little animation of your very highly abstracted neuron model. If you effectively have a series of incoming spikes, then at some point that neuron is sufficiently excited to emit its own spike. We like that because in absence of any activity, the thought is that perhaps you're able to reduce the amount of memory accesses you have, or if you're encoding information over time, then you're able to get a far more compressed and efficient representation of information. What's going on under the hood? So another fun animation to click play on. So on the top plot, you're going to have the input current. If we could hit play on that right-hand plot as well. Excellent. You've got the input current coming in, so you can kind of see the dynamics between the, I guess, intracellular and extracellular mediums. The state of that neuron has some sort of localized memory. A bunch of training hacks that we can adopt from the deep learning crowd if you're just shooting for deep learning problems. And the benefits that you might see from this are sparsity, low precision arithmetic, and compressed information representation. Happy days in a way. Training these can be a pain. So yeah, we spent a lot of time building out this Python library, SNN torch, spiking neural nets, obviously. It's an open source library made for gradient-based optimization of spiking neural nets. It enables a couple of fancy learning techniques. If you know PyTorch, bam, super easy to get the hang off, and it's had a couple of downloads. And it's also compatible with neuromorphic hardware out there, and a few cool places are playing around with it for a few of their own problems as well. A bunch of really rigorously documented tutorials if you want to start from scratch, or if you already know what you're doing, if you're perhaps a pro with modeling and not so much of a pro on the deep learning side, this can help you bridge that gap a little bit. My question to you though is, does this address the right problem? If we get cheaper models, then do we win? The cold hard truth is not necessarily, right? Because if I was to take that preventative action and try to suppress that seizure or suppress whatever neurodegenerative disease that you're concerned with, you're going to break this down into a few different components, right? You've got sensing, right? You're measuring some sort of activity from the brain. Maybe that's done with a neural network to say, yeah, there's a seizure coming, or yeah, there's Parkinsonian behavioral parameters there. You've got a controller that says, okay, we need to activate something preventative. We need to trigger the stimulus. And then you have the stimulator itself. And that is going to form some sort of closed loop feedback in an intelligent system. So what's the cost of all of this? Okay, and I'm going to be very abstract and like high level about this. Low, medium, high. There's approximately a 10x difference where your low might be tens of microwatts, your medium might be hundreds of microwatts, and your high might be in the single digit milliwatt scale. Sensing, around low to medium, right? Control, that can be very low. You don't necessarily need a neural network there if all of that effort has been offloaded to the sensory interface. The neurostimulator, on the other hand, well, in the context of a seizure, you might just want to suppress that at the onset of an event, right? And so that looks like a very bursty workload. For the most part, that neurostimulator feedback is off, but there's going to be times where there's very high power drain, right? And this isn't characteristic of every single type of workload either, right? If you have something that's a bit more continuous, then in that case, the sensing can follow a far greater range. Your controller, likewise. And the neurostimulator, that's constantly on, right? If you're constantly exhibiting some sort of, I guess, behavioral weakness that needs to be suppressed, then you're always going to be triggering that feedback. And so there's different problems mean that there are, I guess, different bottlenecks throughout the whole pipeline. In this case, it's the neurostimulator that's exhibiting the highest amount of power draw, right? Note that there's no neural network there. I could throw a spiking neural network into the sensing, into the controller, and I'm really only addressing like 5% of the total problem. Pain in the ass, right? Like, is all of our group's work kind of done for nothing at this point? Well, hopefully not, right? So we're looking into this paradigm of energy-aware learning. I think this is kind of elegant, right? Because the brain was somewhat self-optimizing given the physical substrate that it had to work with. Can we do the same thing in this context? So let me paint the picture for you. Let's say that we have a cute little rat simulated for now with 80 Hodgkin-Huxley neurons, and someday using Brad's tools, we throw that into a Parkinson's disease classifier, throw that onto some neuromorphic hardware that's running this at the microwatt scale, hopefully, feed that back into a controller that says, yeah, let's stimulate, let's try to suppress these your behavioral problems. Your first reward signal is energy, right? Oh, sorry, it's efficacy, right? Did we do the task that we set out to do? Are we able to reduce the onset of Parkinson's or the onset of a seizure? And your second reward signal, and this is where things get interesting, is energy, and if you care about it, then latency as well, right? So you're literally rewarding your overall model by having a neurostimulus feedback that reduces the amplitude only to what it needs to be. And then there's some benefits there, right? Because you can avoid habituation or excessive energy consumption out of all of that. Now, in practice, you would probably combine your sensing and your controller into one artifact. And we've run a series of experiments that are published both at ISCAS and at this conference by my grad student, Bin. Now, what you're looking at here, you know what, in the interest of time, we don't need to look at this. There's just various reinforcement learning strategies, but I think what's more interesting is here. So on the top plot, you've got the ratio of alpha beta band power, and you want to decrease that below this red dashed line. And over time, your model is going to figure out how to adapt to an individual patient, or in this case, to your rat model. And on the bottom plot, over episodes, over training episodes, you're going to have frequency, amplitude, and my eyesight is too weak to be able to tell you what the other one is. But ultimately, you are able to reduce this by rewarding energy efficiency. We swept across various sparsification degrees with the neural network as well. What that means is that we rewarded the model, the neural network, for reducing spiking activity in the detection of whatever it needed to detect. And yeah, overall, you're kind of getting 20 or so microwatts of gain as a result of this. So what that tells us is the fact that you're using one of these models is good enough. You probably don't need to optimize the SNN itself. Rather, you need to optimize your cyber-physical feedback, right? You really want to be targeting that neurostimulator. Okay? And this goes far beyond neurostimulation as well. So I mean, outside of clinical care, I mean, my lab is very much focused on computation, right? Be it in the context of the brain or outside of that. If we're targeting the workloads that are ridiculously expensive, let's consider a language model that's trained for predicting the next token, the next word. We throw that onto some hardware, GPU or neuromorphic hardware as we do. And your feedback loop is, again, some reward signal that says, hey man, my GPU actually ran this training loop with far more efficiency than what we would have otherwise gotten. And you're able to pass that signal into an agent that can rewrite your code for you, right? So that's probably one of the biggest issues here. Up until now, we didn't have some sort of way to, I guess, provide the amplitude frequency, to turn the amplitude frequency knobs for things that are as complex as language models. So we've actually done some preliminary tests. So in that top right, if you can hit play on that, what you're going to see is some toy experiments on some, I guess, a toy GPU, gaming class GPU. That top right video, if it gets played, is basically, it's the Neuromorphic EMBS data set. There we go. And it's basically an event camera that's just kind of moving and displaying the number 0 in this case. Okay, cool. That's fine. This is just a toy setup. But as we ran this on a GPU, what you're effectively observing is that over time, this system was actually able to find some sweet spot where energy on a GPU was minimized. And this is kind of weird because GPUs typically need kernel level optimizations to get cheaper and cheaper. So what actually happened? Well, we found that this agent basically took a couple of hours to integrate this visual data set that has many, many millions of events occurring in that video and compressing that down to effectively 10 time steps, right? So it just integrated it into larger time bins. And by reducing the amount of time that the data set took to run, that was far cheaper for the GPU to process. Now, with all due respect to my colleagues in neuromorphic computing, it took us about 4 years to come to the same realization. So with that being said, I think that there's a lot of value in taking these kind of more weird and orthogonal approaches to being inspired by the brain. And none of this work is possible without the absolute star students in my lab. In this case, Sky led the work on future guided learning, and Bin led all of the work on energy aware learning. So with that being said, we can chat afterwards and go into more detail about this later. Thanks.
All right, so our next speaker is Gina Adam from George Washington University. Who's going to be talking to us about neuromorphic hardware inspired by the hippocampus, but also towards more compact and efficient hardware for implantable devices.
Hi, everybody. It's really nice to be here, to have made it here to sunny San Diego. I did my PhD in Santa Barbara, and after I left Santa Barbara, when I joined George Washington University, probably the first week, one of our very senior colleagues in biomedical engineering department invited me for a meeting with him. And he said, look, we can put these devices that you're working on, memristors and circuits made on them in implantable devices. Let's brainstorm an idea about that. And I was terrified as a beginning assistant professor about this. It sounded like science fiction, a lot of work on thinking about what we need in terms of computing, but also the sensing and the actuation parts. Exactly as Jason was mentioning, there is a lot in thinking about how to integrate computing with sensing and actuators for organ conformal systems is another example of a potential application. So the overarching motivation for this talk is to think about what we can do from a computing perspective to make this idea a reality. And I want to tell you a little bit about the state of the art of where we are with these organ conformal platforms. For cardiac applications, which my collaborator was working on, we have these fabrics of sensors that are integrated on this conformal heart sleeve. And these sensors can be different types of sensors, from sensing the electrical activity to different pH sensors, and even some sort of actuators, for example, micro LEDs that can apply optical stimulation. And there is a possibility of making these sensors quite densely integrated and to be able to capture the different electrical behavior of the heart and make these maps of the heart activity. And also for the neuro applications, we have similar fabrics of sensors like ECOGs. And there is a lot of very interesting work showing how this can be integrated with the stimulation of the spine. So this is really, really interesting. Of course, it's not my field, but what really surprised me is the necessary capability for computing. You can see here that, for example, for the neuro application, the person needs to carry a backpack to support the computing. And I mean, that's really very difficult for the patient. So the challenge is we need this compact and very energy efficient computing hardware to support the processing of these high number of sensors and of actuators. So in my work, we're working on new types of hardware, in particular novel analog devices called memristors, and they offer performance promises of orders of magnitude, 3 to 6 orders of magnitude better energy efficiency than their digital counterparts. And these devices operate inside with an ionic filament, so they have some synaptic inspiration. And by changing the filament state, we can change the properties of the device, and that contributes to learning in the system. And we can also retain the filament by removing the applied voltages to the system, and that shows memory. So these two properties of learning and memory are key, and of course they're key also as for biological synapses. But on top of that, the memristor devices can also be made very small. I have here an example of a 2 nanometer by 2 nanometer device shown in the literature, and also very, very energy efficient with currents down to the picoamps range. But how can we actually get these improvements in terms of energy efficiency at the system level? So in my group at George Washington University, we have been working on drawing inspiration from the brain to build towards compact and energy efficient computing with a vertical integration across the stack. And I'm going to take you all over this like pyramid in the next slides. Starting with the bioinspiration, and we're working on drawing inspiration from the rodent hippocampus, given that it's one of the most studied regions for learning and memory, and it has a lot of rich experimental data. And my collaborator at George Mason University, Professor Ascoli, has put together an inventory of the different types of neuron behaviors and connectivity patterns from the literature in an inventory. So in the rodent hippocampus, there are 120 different neuron types with different spiking patterns. And our goal is to try to investigate why do we need such diverse spiking patterns, and how can we translate them into a neuromorphic hardware. So moving up from that, we are looking at implementing these neural functionalities in analog circuitry, and I want to give kudos to 2 of my students, Shushmita and Aral, who have been working on this, looking at implementing Izikevich neural model that has been reduced to 4 parameters and comparing it with experimental data for different neuron types at different neuronal ages. And you can see here that we're able to get similar spiking patterns for these different neuron types. And currently we're working on actually implementing this in circuitry. We are fabricating these chips and we're looking at how to expand this to other neuron types. These are just granule cells. We're looking to have other different neuron types with more complex spiking patterns as well. But the neurons is not the only piece. We also need to look at the synapses, and I wanted for, if there are students in the audience, to showcase where I started as a PhD student, not far from here, and working on these member-served devices and optimizing them. So when I started my PhD, the devices that were there in my group were looking like what you see here on the left. The devices were kind of working, but they were blowing up. So my PhD advisor, Professor Dmitry Strukov, he said, well, you need to integrate these devices into arrays, and you need to integrate them, the arrays, three-dimensionally, one on top of each other. And I was like, well, but the devices are blowing up. So a lot of work had to be done on optimizing these devices. You can see here, after 5 years for my PhD, I was able to fabricate these damage-free devices integrated in arrays, stacked in 2 dimensions. And we're also able to measure these devices for their different spiking time-independent plasticity behaviors, showing that different SCDP patterns can be implemented similar to what we see that are different SCDP patterns in neuroscience. So this was very promising, but of course, we only had a few number of devices. So when I moved to George Washington University, our goal was let's make bigger number of devices and start working toward more larger scale. So my PhD students, Shweta and Imtiaz, have been working on this, on integrating these member-served devices on transistor fabric at scale. And we have sourced the circuitry from foundries with the last layer being pre-planarized. And we were able to fabricate our devices in our nanofabrication imaging center and have arrays with 20,000 of these devices. So moving from around 200 devices that I fabricated for my PhD to around 20,000 devices with a yield of around 85 to 90%. And this has really enabled us to think about what we can prototype on top of these devices. So next up, we are able to have these devices packaged and integrated with a custom board that connects with an FPGA board that connects with a computer for a user to actually be able to program algorithms on this platform. And my PhD student Osama and Joey have been working on this, and this is a collaboration that we've been having with NIST and with Western Digital. So recently, we have been able to implement some small spiking neural network on this. It has 3 presynaptic neurons and 3 postsynaptic neurons, and they're connected with each other in a feedback loop. And the activity passes through the member-served devices. Everything is on this hardware platform. And you can see here that the presynaptic neurons send activity, and the postsynaptic neurons are reading that activity and sending it back. And they're able to influence each other going through the member-served synapses. But this is a very small model. And right now we're looking at scaling this up, going up the pyramid scale into having a more biologically realistic hippocampal model. We're looking at the C3 subregion and scaling down that from a full-scale model with around 90,000 neurons all the way to a much smaller model. Right now, we have a model with around 175 neurons that we're optimizing and reducing the number of neuron types from around 8 to 3 neuron types so that we can implement them on this member-served prototyping platform. We're currently optimizing this for realistic periodic behavior, but the results seem very promising. But ultimately, we want to utilize this towards practical applications. And I want to give a shout out to my students, Jolene and Lei, and also to my senior collaborator who inspired me, and I mentioned at the beginning of my talk, Professor Igor Efimov, who right now moved at Northwestern University. to look at these memory serve-based neural networks for smart cardiac implants and taking actual data from human tissue and identifying abnormal wavefront in these tissues, and being able to apply these algorithms to terminate arrhythmia. So if the video can be played, you can see here right now we are in simulation, but you can basically see here that by applying computing where this abnormal behavior is and applying only locally the actuation for therapy, we're able to terminate the arrhythmia. So our vision for the future is to integrate these memory stiff hardware into chiplets and incorporate that with sensing and therapy for such smart implants of the future. With this, I would like to conclude saying that we need sensing, computing, and therapy to work together, and emerging analog neuromorphic hardware can help with that. Thank you very much.
All right. So our next speaker is Dr. Julia D'Angelo from the Czech Technical University in Prague. She is also the co-host and co-creator of the Brains and Machines podcast, as sponsored by EE Times. Please give it up for Julia D'Angelo.
Thank you, William. Hi, everyone. So I'm very happy to be here today, and thanks for having me. Yes, I'm Julia D'Angelo, and I just became an assistant professor, so I'm way younger, let's say. Yeah. So I'm very nervous today, but let's see what I can tell you. So I will talk about what's catching your eyes and why we look in certain places and why we do not look in other places, right? Through the lenses of event-driven sensing and neuromorphic computing. Let's go. Now, this is the dream of everyone, right? You're having a meeting, and then there is a robot that is preparing a cup of coffee for you and is cleaning and everything, right? And we want this robot to perform this in an unconstrained environment with low power consumption and, of course, low latency, because we need them to react very fast, right? But is this happening? No, the best we have is a vacuum cleaner, right? So we are hoping to get something a little bit more complex, like what the little cat is doing or the little fella that is interacting with the object. And in this very moment, I'm anxious, I'm nervous, I'm standing thinking what to say later on, and my brain is consuming the same energy of a light bulb, right? So this is a thing that it's really nice to say all the time. And this is my motivation to why using neuromorphic sensing and computing, because what I want to do is at the moment applying it on robotics for milliseconds and milliwatts power consumptions. Fine, let's go back to our problem. Now, we have the coffee is cleaning, but something is happening, right? My cat is about to burn. So how do I tell the robot, just forget about everything else and focus on the candle? How do we define attention? It seems easy, but it's not true, right? So at least I'm trying to respond to this question from many years. Okay, so what we see here is a person that is typing on a keyboard, and then something is happening, the phone is ringing and is responding to the phone. If only these were not two completely people. Okay, complete different people. And this means, if you haven't seen it, is that your brain is actually functioning as it should. We do not focus on details, but we rather try to understand what's the most informative information in the scene, which implies, in this case, do not even focusing on the fact that these are two different people and getting all of the details. So this means we do not process everything and we make up lots of things. Let's go on. So, problem of before, we have this robot and we need to make the robot attentive. This creepy guy, by the way, is ICAP and is the humanoid robot coming from the Italian Institute of Technology. And yes, I come from the same land of ICAP. And let's think about what we do, for example, in our brain, right? At the start of my PhD, I started focusing on the secondary visual cortex. And there we have some very nice cells that are called border ownership cells. What they do is basically perceptually grouping the information, right? So we have a bunch of information in front of us when we look around, and we perceptually group the information to understand something. And then thanks to Russell et al., which et al. Means also Ralph Whitty and Cummings from the Johns Hopkins University, came out with this nice model, which is basically extracting different features, intensity, color opponency, and orientation. And then on top of that is doing or performing what the border ownership cells do, which is, you can see there the border ownership cells and the grouping pyramid. Again, grouping the information together to make sense of it. So what we want to do here is to try to understand if there are areas in the scene where we should pay attention. Let's go on. So for my robot, what I thought that was important is to use the 3 different channels of information, which is depth, because I want the robot at some point to do something. So kind of get something there or grasp something there. Motion, because it's inherently interesting. We are always very alert to things that are moving, specifically towards us. And intensity because it comes from the cameras that I'm using, which are event-based cameras. So this is what I managed to do in my PhD. So you can see that we have the intensity, the depth, and the motion channel. On the left, you can see the events and the saliency map. The saliency map is basically what is salient in the scene. And you can see that it's seeing the object and paddle now, but it's not seeing the line there. The line is not recognized or detected as an object. On the right, you can see, I cannot point to you there, but you can see that in the green screen, there is the red circle is ping-ponging between the 2 objects, whilst instead, if you input the disparity information into the model of attention, you are focusing on the closest object to the robot. And this is because at some point you want the robot to grasp the object, and you need to tell the robot, okay, this is the object, otherwise it would be always in between the 2. The third one is the motion. And if you know the Reichard model, that's it. This is the Reichard model. And basically this is done on a Spinnaker as well as the intensity channel. So the intensity channel has been done on GPU and on Spinnaker too. So of course, I have a bunch of quantitative and qualitative data and results that I cannot explain. What I can tell you is that most of the time, when I do saliency-based models, I need to compare with real human subjects looking at a scene, right? So imagine having a very dumb bottom-up model, and you need to compare your results with a real human being. So it's not easy. But what I can tell you is that our event-based approaches, instead of the frame-based one, because of the sparsity of the events, they're very localized. So it's slightly better in comparison to human fixations. Okay, let's get out from here. And this is actually what is, I think, the most interesting thing, right? At some point, I started benchmarking against myself, basically. And you can see that the intensity channel, if it runs on a GPU, runs in 100 milliseconds. If it runs on a Spinnaker board, it runs in 16 milliseconds. How many Spinnaker boards you need to do so, that's another story, and I'm not going to address it now. But on the right, you have, for example, the depth channel that is running in 170 milliseconds. This is because we need to extract the disparity information before, so we need 70 milliseconds, around 70 milliseconds to do that, and then we perform the attention. But what is really interesting, and I always love, is the motion channel, so the Reichard model that needs and will give you the information of direction of motion in 4 milliseconds, which is, I don't know, I'm still happy about it. Let's go on. Okay, so this is visual attention. At some point, I got very interested in what's happening in the retina. The retina does so many things that I wasn't aware of, and I wanted to make aware too. So these are all of the main cells that we have in the retina, and I focused on the amacrine cells. The amacrine cells basically are already performing some sort of object motion segmentation for for us. And we do not need perception for it. And that's, again, very happy. Either I'm easily excited or this is actually very, very cool. So I started from this biological data. And then I wanted to perform the same model, so to implement the same model with event-based cameras. So if you look at the video, you can see that what I'm doing, I have basically the event-based cameras on my leg, and I'm kind of like anxiety moving my leg, right? And you can see the static and dynamic events, right? Because you can see my hands moving, but also the static events. What you do with the amacrine cells or this model is the ability to completely disregard the static information. And how you do so is looking in coherent and non-coherent motion in locality. So of course, this is very cool because I'm already dropping 85% of the incoming events that if you work with event-based cameras, is a pain to process lots of events, especially when the camera is in motion. Then another thing that, of course, I had to look, it was, okay, I'm dropping events. Am I doing it reasonably? Yes, it came out, yes. I don't have time to look at those quantitative data, but please drop me a message or please stop me and I will tell you more. So I have done a bunch of benchmarks there. Let's go on. So then what I wanted here, it was a complete closed loop architecture, right? So what I wanted was, okay, I have the SPAC, which is, I'm just using the events from the camera for now. So I'm getting the SPAC on a pan and tilt unit. Then I'm jittering the pan and tilt unit, performing microsaccades. Then after those microsaccades, I distinguish, I do object motion segmentation. Then I perceptually group the information, I do attention, and I get a point that is a point of salient point, which is where I should pay attention. So I get the point, I throw it to the control, and then I saccade. So I perform the eye movement to then place that point of interest in the center of the retina. Now, what is actually interesting here is that if I do not perform these specific movements, so microsaccades, the object motion segmentation does not work at all because I need locality, right? So here it's the proof that everything matters, and it's kind of like what I'm trying to push in terms of embodiment, right? The algorithm is specific for the body and vice versa, right? The body and the movements, everything matters here in the implementation. So what turns out is this, right? You can see that the algorithm, it understands what is in motion in the scene and is placing that thing that is in motion at the center of the visual field. You can see the pan and tilt performing microsaccades and saccades. Another thing that I'd like to say is that what I like here is that at some point I would love to stop processing the information whilst with saccade. I do not think that we should process all of those events while we move the camera. Now, another thing that is really interesting is that if you run the model on my M2, it takes 120 milliseconds, but then I just recently finished the implementation entirely on SPAC, and it's using 1 millisecond and less or equal 1 watt, right? So it's kind of like, promising. Let's go on. Fine. So this is, of course, part of a bigger problem because it's just how we move our eyes around. And what I'm actually doing and what I'm interested in mostly is trying to understand how infants understand and perceive objects. What infants do most of the time when they're seeing for the first time an object, they visually explore it, right? So using eye motion, so using visual exploration to get the object perception. So this is what exactly I'm doing, and I'm basically using sensory motor contingencies, so linking actions with the sensory information through time to create then the concept of objects. I'm doing it in a different ways, and is basically the project that my PhD, Sharka Liskova, is continuing now, which is, of course, by inspired active vision through the lenses of visual exploration. Of course, I do other things, and these are the other projects of my students. And for example, there is another nice implementation of shoreline and coastline detection on Spinnaker. Again, this I'm very happy about this because we used the Spinnaker 3, which it has only 4 chips. So we used around 20K neurons, and it responds with a very good accuracy, over 85% in 20 milliseconds with 0.3 milliwatts for shoreline monitoring. Then what I'm doing is also visual stabilization. So trying to use those microsaccades to increase the confidence of the recognition of an object, because it is exactly what we do. I'm also doing drowsiness and sleepiness detection via saccades, motion direction detection on top of the object motion segmentation, and keyword spotting to influence where we move our eyes. All of this and all of my models tend to be very lightweight, which is the reason why I'm starting to explore visual prosthetics. I know nothing about it, so you cannot ask me anything. I'm just exploring it. So I'm starting it now, and there is lots of things that, of course, can be said. And I need to understand if this could be done with subretinal circuits or cortex circuits. I need to understand everything, but this is where I hope that my future will be. And of course, thank you so much. These are the university and the companies I collaborate with the most. Please drop me a message on LinkedIn. You find me there. And of course, yes, I do have a podcast. Please go there because there's a bunch of very nice people there that you should listen to. It's the Brains and Machine podcast together with Sunny Baines, University College London, and Ralph Etienne Cummings, Johns Hopkins University, and also Sottosoglia, which is subthreshold in Italian. I'm very proud of that too, with my lovely Fabrizio Ottati. And thank you so much.
And now time for our last speaker, Dr. Elisa Donati. We've seen her work before earlier in this conference, so I don't think she needs an introduction, but let's hear about what she has to say.
Hello, thank you. Thank you for having me here. I'm the last one. So I'm going to give you some example of how we can use neuromorphic for a specific case that is the one of prosthetic control, and in particular for upper limb. I'm not going to go into motivation of why I do that. First of all, because I just want to do that, so it's fine. And second, because, you know, I have silicon neurons and synapses. And when I started to work in the field, I was thinking, oh my God, I want to interact with the body, I want to interface with the body. So why not doing something like using neuromorphic? And I also don't want to tell you what is neuromorphic for me, because usually when I start the presentation, I always need to start with the definition of neuromorphic. But yesterday we had the presentation of the keynote, it was Giacomo, so you know what is neuromorphic, I do exactly the same. And actually in particular, so let me go for the first, specification of my presentation. My approach is slightly different from what we saw before. For example, I don't want to do something large. I actually want to do something very small. And I want to do something that is, implantable, embeddable, something that we can put at some point in our body. And for doing that, I need to build something that is very small and something that it cannot fully rely on learning, because unfortunately, when we work with the body, we don't have a lot of data, so we cannot teach our network to learn something. So what I do is something rather small and something that doesn't fully rely on learning. And now I'm going to go more concretely in some example. So again, so I want to start talking about this bidirectional upper lip prosthesis. And if the resolution is weird, sorry, because the presentation is supposed to run on my computer, so usually it doesn't work in other setups, but still. So when you want to, replace like an arm or like a hand, so of course you need to generate some movement. So you need to read from the body, but you also would like ideally to send something back to the subject. And this is for many reasons. So once because you can improve the control itself, but also because of course the person can feel, a person can accept better the prosthetics. So you would like to have this bidirectional system. For doing that, we have multiple stages, okay? All of them, they're kind of very difficult and a problem per se. So we can have something like, decode information from multiple sources because, it's not only the muscles, but it can be the muscles, it can be the nerves, it can be the spinal cord, it can be something directly from the motor cortex. So you can really take this information from multiple sources. Then once you generate the control and you want to encode some information about the feedback, you need to build the sensors. That's another problem. And you need to, how can I translate the information of the touch in something that is meaningful for the body? So you need to do something that is like the encoding. And encoding can be biomimetic, not biomimetic, it doesn't matter, but you need to do this stage. And then when you think of when you stimulate the subject, especially if you do something that is peripheral, you unfortunately have only few points of contact. So you need to compress a lot this information. And then you need to understand what is the effect that you have in the brain. So there are multiple of these pieces. And today I would like to show you at least a couple of them. I don't know if I have time, but let's see. So first thing, so we want to decode some information to control the prosthetic devices. So traditionally, everyone knows that, what is gesture recognition. So when you want to recognize something that you do with your finger. However, if you think of how our hands move, and especially for me, that I'm Italian, so you can clearly see how I move my hands. We have an entire language that we can speak with our hands. So it's very important that you understand all the single movement. And this is not that it's just a recognition, right? This is something that is regression. And if you want to map this information from your activity, you need, for example, to map something that is from the muscle activity to the finger movement, so to the finger force that you generate. This is something that is continuous, right? It's not discrete, like just a recognition. That's why first of all, it's more natural. So it's how we move our hands. And second, is also something that comes more natural when you work with neuromorphic engineering or with spiky neural networks when the time is there. So you have the continuous information on time, in time. So that's why I started to move away from just a recognition and moving more towards the regression. And here you can see, for example, that all our fingers are basically an independent problem. So you can control them independently. And when you combine the movement of all of them, you can have the full gesture. So what I'm going to show you now, it's rather simple, okay? And it's also sometimes even annoying because people, when they see this example, they say, oh God, this is a toy problem. It's very simple, but it's actually not. And I can explain you why. So what I did here, it was something that it was running on a microcontroller on ST32, so something very simple that everyone can have for $10. You can buy it, you can implement everything. But I'm showing this example because I want to explain you why I'm using spikes and why spikes are cool. So here, what we had, it was like a 16 channels on the arm or the activity. So we were recording the activity of the muscles. And then what we were doing, we were converting the signal into spikes, because this is something that it's very important if you want to work with neuromorphic, because neuromorphic intrinsically process spikes. So, and more often, all the information that we come from the environment, they are not on spikes. So we need to actually convert the signal into spikes. And there are different ways of doing that. The one I'm using here is rather simple. So I have a bank of filters, so it's similar to what the cochlea does. So I look at the frequency of my EMG, and then I convert the power of each of those channels into spikes. And then I was using those spikes as input for a very simple classifier, like a linear classifier. Sorry, not classifier, a regressor. So in particular here, we were using 4 bank of filters each electron. So for a total of 64 inputs to my network. So in the first stage, and I don't think I have a pointer to, yes, I have it, but it doesn't work. So basically what happened is that you actually have the first phase. So you have the signal, it goes into a neuron, the neuron generates some spikes. And then after that, since I want to use a linear regressor, is not ideal as a first step, but yeah, so I need to convert back into the continuous signal. So I was then using a kernel, if you're from the neuroscience field. And in our case, it's basically I was multiplied by another neuron. So I was actually looking at going back to something that was continuous, this exponential kernel. And then I was implementing my linear regressor. So here is this first stage, the one of the amplification. I forgot about that. So then if we look at the performances, it's a linear regressor. So what we got, this is also something that was in the state of the art. So we got the performance that it was like around 8 of error. So if you think of your movement of your finger is like an error of 8 degrees. And then you can have something, however, that if you implement on a very simple ST32, you can have something that is quite powerful compared to any other kind of state-of-the-art RISC-V device. So you can see here that the memory was lower, 10 times lower, the number of operation was 10 times lower, and anytime I was getting any information, any cycle of the clock. So it was a very fast, so the best latency that we could have. But then the reason why this system, although it's so simple, it works, is because we were using spikes. So if you look at the performance here, if you look here, so you can see that we were calculating the SNR before and after the conversion into spikes, and then the use of the kernel, and also the linearization of the problem. And you can see that using the spikes and then using a kernel, it was making the problem much more better in terms of SNR. So the problem was improving in terms of signal, and it was also more linear. So this is something that made the problem so simple, but because it was so effective, but it's so simple because of this reason, because we were using spikes. And anytime I present this, I get people saying, oh no, but this is a toy problem. And I get annoyed every time. So then I started to do something that it was really deep learning. So I was using all the possible networks that you can think of, transformers with dual modular attention, TCM, CCM, whatever. And I have the results at some point in the slides. So you can see that the system is basically performing better than those methods. So the error, it was slightly higher, so it's 8 degrees versus 6. But still, in terms of other performances, it was better. And I bet you to try to understand, if you move one finger, if you can see the difference between 8 and 6. From the clinical point of view, it's impossible. So there is no point of trying to optimize such parameter if you don't even perceive them. So then, since I was using this hybrid system where I was working something that it was between spikes and no spikes, so a kernel, I decided to move on something that was fully spiking and fully on chip, and in particular on neuromorphic mixed signal chips. So here, what we did, we were performing the linear regressor directly into the chip. And for doing that, we were using population of neurons. So in particular, so we were looking at the movement, again, of the fingers, and we had the massive number of electrodes, and some of them, they were looking in one direction, so the flexion, and some others for the extension. And so then we created two very simple population on chip, something extremely simple. And it was one population for the extension and one population for the flexion, and we were activating that. And in particular, we were trying to, in this particular case, we were suppressing the activation of the flexion because if you look at how we move our hands, we have more activity when we do the extension. So it was basically dominating everything. So, and what we did, we were trying to map, again, this linear activity of the fingers by using the chip. And this simple network, it was actually performing like a linear regressor. So here you can see the activity. I'm sorry that I cannot use the pointer. Maybe you can see that. So these colors, they were the different grids that we had in the electrode. So different parts of the activity of the muscles. And this is valid for the thumb, index, middle finger, and the little finger. So then we were computing this linear regression on chip. So we had something that it was, so you can see one for the flexor and one for the flexion and one for the sensor, and we were then combining together. And you can see the comparison with the linear regressor. So you can see here the activity. So then we calculated the difference between these two systems, and you can see that actually the dynamics is performing very similarly to the one of the linear regressor. So this is, everything is on chip. We were using few neurons, like 8 neurons per population. But still, you can see that the performances, they were very comparable. And in some cases, when there was a minimal number of input, even better. So you can see that they are very comparable. And you can see that also the power consumption was extremely low. It was 7.73 microwatts during inference, of course, for a total of 14 neurons with a mean firing rate of 30 Hertz and 18 Hertz for deflection and extension. So very powerful. So now, so these are two examples of how we can control. So we can generate something to control the prosthetic devices. But since they asked me to do something in the closed loop, I go 1 minute also on the what we can send something back to the subject. And for example, I want to show you how we can simply do some signal compression that is very complicated problem, still using our chips. So we saw some presentation from the days before that our skin is full of sensors. We have many sensors, so they are everywhere. So touch sensors in particular. So we have some Meissner corpuscular, Merkel, Pacinian, all of them, they just get different information. So there are simulators, very biometric simulators that can, once you do some pressure, you can convert the information into those spikes. But then if you are very good and if you want to do something very precise and something that is very similar to what we have in our body, you end up to having this massive amount of inputs. So you have sensors everywhere. So now once you have all those inputs and you can think, okay, now I want to do some simulation on subject, how can I do that? So you don't have all these points of stimulation. Actually, you only have maybe some residual arm. So you would like to stimulate in 1 point, maybe 8 points. So if you're lucky. So the idea is to massively compress this information in much less. So what we did, it was very simple again, but very effective. So we were actually implementing something similar that we have in the nervous system. So in particular in the spinal cord at the level of the chip. So we were sending all this information in 8 populations that we have in the chip, simulating some synapses that we have before getting to the thalamus. And so then we were actually doing that. It was very simple. So we had like 8 neurons per population. So we had 8 outputs, each of them a population of 8 neurons, and each of them, they were kind of implementing a soft Winnet call. So meaning it was kind of slightly inhibiting the lateral to make the signal more clear, to actually have this compression on chip. And you can see it here that we tried this compression with different types of input, and something that it was using 1 finger at a time, something that it was simultaneous. And you can see the output of the simulator here. So with a very high number of neurons, you cannot see that. And then you can see the output of the chip. So the output of the chip, it was actually very following the one of the input. And if you look at the firing rate, you can still see there's some information, temporal information from the original cells. It was still there, it was preserved. So although we compressed, we still have some information about the temporal system. So this was a very short, but just to give you an idea that with the chip, with the same chip, we can do both. So we can do something for the decoder and something for the encoder. And with that, I want to thank you, all my collaborators, all the students, and all of you that are here and listen to this presentation.
Okay, so now we're going to take some questions. Actually, if I could have the speakers go back up to the front, we're going to do questions from the group for the group. We also have a couple of people from the NIH on Zoom who may also pipe in and offer a question. Wherever you want. All right, so who has a question?
What do we do?
Let's go broad until someone far smarter than me offers a question. What's the most proximal bottleneck and what's the most promising way of approaching it beyond what you've shared already?
The bottleneck, I mean, I think if you asked for the neuromorphic field in general, we'd all probably have a different opinion about what the bottleneck is. But I'd say the challenge, we're getting to a point where we can actually build systems, whether really small scale or big scale. But, the number of people who've actually used a neuromorphic system, you're looking at probably like just under 1% of the people who've ever programmed a neuromorphic system on the stage, right? It's just not the technology hasn't gotten into the hands of the community as a whole yet. It's a software issue. In fact, Jason's tools, I think, that's the bottleneck, is we need more SNN torches and things that get the technology into people's hands. I think that's, in my opinion, the biggest hindrance right now. The technology is increasingly there from a hardware point of view.
Yeah, great question. I would add on top of that there is a lot of complexity and diversity in biology. And depending on what application we're going to tackle, it might require somewhat different hardware solution or neuromorphic solution. And doing that optimization for the application itself is, of course, time intensive, resource intensive. So that would also be a bottleneck, trying to figure out what the best trade-off is in terms of providing the best optimization for the problem and going to something that's still resource effective and more kind of a general solution.
And if I may add something, something I was telling a couple of days ago, and it's something, in my opinion, very funny and very practical. So especially in my case, where I want to build devices that I can really implant at some point of my life. So recently I was talking with some people from the FDA approval. And, you know, when you talk about neuromorphic and what the neuromorphic can do, we always say, oh, neuromorphic is cool because it's not only low power, but you can also have something that is able to adapt by itself. So it can change based on the context, based on the input. However, this is a huge problem from the FDA approval. So they cannot approve something that you don't know how it's going to change, how it's going to, you know, evolve in time. So this is a problem. It's a real problem. So if I think of, you know, opening a company, a startup, and doing something like a real device, it's going to be a huge deal to get some kind of approval for neuromorphic.
From my point of view, instead, I'm basically a user of different hardwares, the Spinnaker, Loihi, and etc. For me, the problem is that all of them are different, and it's very difficult to make them run. Like, for example, the spec solves the problem of having a camera connected to the processor, but the problem is that I had to trick because I can only use integrate and fire there if I want to run it on the spec. Loihi, I've never made it run. So I honestly have no idea. To me, it's too flexible for no reason. And it doesn't really make sense for me when I tried to code it. Spinnaker never failed me, but most of the time I had to use really the minimum was 6 boards because I use Vision, right? So for me, the bottleneck is still trying to understand the best hardware that I can use for what I do. Plus, of course, the bottleneck is when you move the cameras around, then you need to deal with a number of events, which I don't know. I think I will get towards the hard approach of not processing them at all whilst I move the camera and just processing in glimpses, which makes sense to me, but also the resolution of the camera, which I really don't think we should get a very high resolution of the camera. We should rather need to visually explore, but this is my approach.
Move on to the next question.
Sure. So I think all these talks highlighted a wide variety of problems you're trying to solve, approaches, different tools. And so can you maybe talk a little bit about how can you make this easier for new people to join in who may not have the specific domain knowledge to figure out what's the right solution, or if there are like easy entry ways to get into bringing neuromorphic into their work? Not a specific question, more of a issue.
Can I start since I have the, so there are many places where you can actually start. One of it is Open Neuromorphic, for example, is you just join the Discord channel and there is a bunch of nerds that will help you for sure. So that's actually the first, step. But of course, like if you ask there, but also Giacomo is basically preparing like a huge service where you can, talk to people and interact with people. So actually, I think it's by now, if you're just alone by yourself, not talking to anyone, you can follow tutorials, but it's better to talk with people that has done these things. Like if you ask him about SNN Torch, it's going to be easier. There is a very nice network, I think, of people that will help you. So just join those channels. That would be an advice. Yeah.
Yeah, I mean, I'll flip your question around a little bit. The neuromorphic field is pretty small still. And we, yes, it's kind of high activation energy to get into the field, and it's complicated and it's different. But that said, because it's a small field, we need all of you, basically, right? I mean, everyone is an expert in what they know, and the neuromorphic field needs that as well. And I think most people in the neuromorphic field come from other communities, right? And the field has been built by people coming with their expertise in neuroscience or electrical engineering or physics or comp sci or wherever. And I think we just, the field's at a state. It's not like we all have the answers up here. We're just not telling you. We're all learning the answers together. And in this sort of, thing that Julia's talking about, these sort of online communities are growing. There are a handful of conferences, Telluride, Kapakacha, NICE, Icons. They're, just start jumping in, right? We want more people involved.
We have time for one more question. See you hand over there.
So, I mean, we know that neurons are not Hodgkin-Huxley machines. We know they're not leaky integrate and fire machines. And, it's not even totally clear that all the neurons that we think spike actually spike, and we don't know that the learning rules are coming from spikes necessarily, and it could be a totally separate system altogether. How do you square the like march toward larger and larger neuromorphic simulations or neuromorphic technology with like, maybe this isn't even where learning rules come from. Maybe this isn't even the the sorts of like compartment models that we should be thinking about to move forward towards something that looks like the brain.
I'll answer, then I'm going to hand it to Gina because she'll have her answer on this too. But I think you're right. I mean, there's a lot about neuroscience. There's a whole meeting next week of different opinions about what neurons are and so forth. And I think the field's rapidly evolving. Dendrites right now are really trendy. Dendritic computation. I do stochastic synapses. The current neuromorphic hardware doesn't really look at stochastic synapses. So I think that it's a co-design. Like that's the word of the decade and for neuromorphic.
Yeah, I would say the same thing. Co-design is really where it's at. But I'm an electrical engineer, and when I look at neuroscience, it's honestly frightening. There's just so much complexity, so much stuff happening that can be paralyzing. And like, yeah, I mean, how do we even start to tackle this? But I think the neuromorphic community has done really well in taking the first steps towards understanding what our contributions might be and trying to distill that information. We're still at the beginning. So as Brad mentioned, we're welcoming of people with different expertise to help us understand, and hopefully we're also contributing with the understanding.
Now, I think that there is a time and place for that deep in neuroscience inspiration. But if I can kind of be our own devil's avocado for a second here, it could be the case that the brain has a particular neural code because of the constraints of the brain, the physical substrate that it was limited to. Now, if we're going to be building brains in silicon, we do want to be quite self-critical here and ask, does it make sense to integrate the most biorealistic neuron model ever and just pray to someone that you're going to get some sort of emergent property? Now, perhaps in some cases, when you're building out a large-scale connectome, you do see something that does matter. But if I can inject some of my engineering bias here, now I'm very clearly quite far from a neuroscientist myself, chip designer, software engineer. So I'm looking at quite arm's length principles from the brain to the point where it actually pisses off a bunch of people for me to call myself a neuromorphic engineer. They're like, is that really neuromorphic engineering if all you're doing is taking advantage of sparsity and efficient information compression? Maybe, I don't care, doesn't matter. If it works, then I'm quite happy. So there are things that don't necessarily make sense to integrate on modern technologies. But then that goes back to your co-design issue, right? So design something new at the hardware scale if it makes sense.
Let's thank our speakers one last time. I believe there's a break, so you can go check out posters in the other exhibit.
Thank you, Will. Great job.